Hi, my name is Noah Dickinson, and today I'll be discussing a project I've been working on which investigated the role of prostaglandin signaling in the development of radiation-resistant forms of breast cancer. So, I have no relationships with for-profit or not-for-profit organizations, and financially this project was supported by the Natural Sciences and Engineering Research Council of Canada, the NSERC Discovery Grant, the Northern Cancer Foundation, the Northern Ontario School of Medicine University Faculty Association Research Development Award, and the Canada Graduate Scholarships for a Master's Program. Previously, our lab has used MDA-MB231 cells as a model for triple-negative breast cancer, and we've actually generated radiation-resistant variants of these cells. And one of the interesting things that we had found was that the prostaglandin signaling system was very heavily dysregulated in these radiation-resistant variants. And so what we aimed to do with this project was we wanted to look at specifically PTGS2, which was had a 14-fold increase in expression in those radiation-resistant variants. And we wanted to look at if we overexpressed PTGS2 alone, if it was going to cause that increase in radiation resistance that we had observed in those cells before. And so we used a CRISPR-Cas9 overexpression system to overexpress PTGS2 in these cells. And we confirmed that, yes, in fact, we were overexpressing PTGS2 using RT-qPCR and Western blotting to confirm it at the mRNA and protein level, respectively. And we went on to do a clonogenic survival assay post-irradiation to try to confirm what effect was going on with the radiation resistance. And interestingly, we saw the actual opposite effect of what we were expecting to see. Whereas before we saw the radiation-resistant cells had high PTGS2, when we caused the increase in PTGS2, we saw that the radiation resistance was decreasing. So this is the opposite of what we were expecting, and it kind of tipped us off that maybe something else is going on in terms of cancer progression, or maybe the PTGS2 overexpression alone wasn't enough to cause the actual radiation resistance that we had previously observed. And so we wanted to look at other factors affecting cancer progression. And so we looked at cell proliferation, and we found that there wasn't actually any significant effect on cell proliferation. We looked at adhesion to extracellular matrix proteins, and we found that, interestingly, fibronectin, adhesion to fibronectin was significantly decreased for the PTGS2 overexpressing cells. And so we further wanted to investigate adhesion, so we did a soft agar colony formation assay where we plated the cells in a 0.5% agar media layer. And this essentially just removes any sort of basement layer that the cells would normally bind to, and since they're epithelial cells, they need that layer to actually grow properly. And so we actually found that the PTGS2 overexpressing cells were actually they showed significantly increased anchorage-independent growth compared to the typical MDA-MB231 cell control. And so overall, the conclusions to kind of draw from this, PTGS2 overexpression alone doesn't seem to cause radiation resistance to increase like we had previously expected. And additionally, it doesn't seem to have an effect on cell proliferation, but it does have an effect on cell adhesion, particularly to fibronectin. And when you increase PTGS2 overexpression, you actually end up with increased anchorage-independent growth. And so future progress in this project aims to actually investigate overexpression or knockout of specific downstream products, things like prostaglandin E receptors 2 and 4, which those are downstream from PTGS2 and are a little bit more likely to actually cause a more specific effect. And thank you for your time, and thank you for checking out my poster. Have a great day.